Hello friends, welcome back to my YouTube channel Nastin Padasalai. This is a part 11 video. Currently we are revising for our GATE examination. So first we'll be studying protozoal disease. Malaria is caused by Plasmodium and it is transmitted by the bite of mosquito. Leishmaniasis is caused by Leishmania and it is transmitted by the bite of sandfly. Next is African sleeping sickness which is caused by Trypanosoma brucei and it is transmitted by the bite of tsetse fly. Next is Chagas disease which is caused by the Trypanosoma cruzi and it is transmitted by kissing bug ficus. Okay, so the other name of this kissing bug is triatamine bug. Next, we'll be studying parasitic worm diseases. So, first disease is the schistosomiasis. It is caused by schistosoma and the transmission is by water. Filariasis, which is caused by Wucheria bancrofti and it is caused by the mosquito bite only. River blindness, which is caused by Onchocerca volvulus and it is transmitted by this bite of this black flies. Okay, so you should know what are the transmitting vector responsible for causing the protozoal disease and parasitic worm disease. Hookworm infections is caused by Nectator americanus and Ascaris is caused by Ascaris lumbricoides and the transmitting agent for both hookworm infections and Ascaris is soil. So the next we'll be studying about the disease transmitted by arthropods. So these are the list of disease and these are the arthropod which will be acting as a transmitting agent or vector. First disease is mosquito. The disease transmitted include malaria, filaria, and viral encephalitis like Japanese encephalitis or viral fevers like dengue, West Nile and yellow fever, dengue hemorrhagic fever. Next is housefly. The arthropod responsible for the transmission of diseases include typhoid, paratyphoid, diarrhea, dysentery, cholera, amoebiasis, poliomyelitis, conjunctivitis, trachoma, anthrax are caused by this particular housefly. Sandfly, the disease transmitted by sandfly include Kala-Azar, oriental sore, sandfly fever and Oria fever. Okay. Next is tsetse fly which is responsible for transmitting sleeping sickness and louse which is responsible for transmitting relapsing fever and pediculosis. Next is rat flea which is responsible for causing this particular plague and black pill is responsible for causing river blindness. Okay. Next is red dew whip bug which is responsible for causing Chagas disease. So that's why I had mentioned over here. So if you check, Chagas disease is caused by Trypanosoma cruzi and it is transmitted by this kissing bug ficus or which is also called as triatamine bug. So here you can able to find other name. It is also called as redouit bug. Okay. Next, heart tick, which is responsible for transmitting tick typhus and viral encephalitis, and also some other hemorrhagic fever like this particular Kayazunner forest disease, or it is also responsible for causing tick paralysis disease also. And this particular heart tick is also responsible for causing tularemia. Okay, next soft tick which is responsible for causing Q fever which is also called as query fever and relapsing fever. Okay, next is with regarding to this mite which is responsible for causing the scabies, and cyclops which is responsible for causing this guinea worm disease and this fish tapeworm. So cyclops is just a variety of this crustaceans only. So at last cockroach which is responsible for causing this enteric pathogen correlated diseases. Okay, so the next we'll be studying an important topic, that is culture media. So first type is complex media. So what are the characteristics of complex media include? It is generally composed of ingredients like peptones or natural extracts. So these particular chemical compound may vary in their chemical composition. Okay, next is chemically defined media. As the name suggests, this particular media composed of precise mixture of pure chemical like ammonium sulfate. Okay, third type is special media, and these are the subtype like enriched media, enrichment media, selective media, indicator media, differential media, sugar media, and transmute media. First is enriched media. So this particular media generally used to grow bacteria which are more exacting in their nutritional needs. So in the basal media, they will be adding substances like blood, serum, egg. Okay, next is enrichment media. So this enrichment media is similar to your selective media, but generally this particular media is designed to increase the number of desired microorganisms to a detectable level. So what is mean by selective media? Selective media means suppression of unwanted chemical, at the same time encouraging or promoting the growth of desired microorganisms. So there will be a employment of selective agent. That particular agent will be promoting the growth of only desired microorganism, at the same time stopping the growth of unwanted microorganism. Next is indicator medium, which will be having an indicator agent, and this particular indicator will be changing the color once the bacteria start to grow in them. Next is differential media. So this particular media will be having an ingredient that can be changed by few bacteria in a recognizable way. Okay, so based on the colonies you can find that particular microbe. Okay, next is sugar media. So most commonly sugar fermentation can be employed in this particular sugar media. Transport media, a transport medium is just a holding medium which is designed to preserve the viability of microorganism. At the same time, in this transport media does not allow the multiplication or growth of the microorganism, just used for transport purposes only. Last fourth type is reducing media, which is used for growth of obligate anaerobes. Okay, anaerobic bacteria. So these are the list of liquid media currently employed. First is peptone water, which have peptone, sodium chloride, water, and the pH will be 7.4 to 7.5, and it is used for carbohydrate fermentation. At the same time, it is also used for some other biochemical tests like formation of indole. Next is nutrient broth, which is made up of peptone water and meat extract, and it is used for routine culture. At the same time, this medium will be used for a base for preparing for some other culture media, and while studying growth curve, you will be employing this particular nutrient broth. Glucose broth means nuclear broth. In addition, they will be adding 1% of glucose, and it is used for blood culture, and this is also promoting the growth of many luxury growth of many organisms. Next is enrichment media, and it is classified in three types like tetra broth. Okay, next is selenite F broth. Next is alkaline peptone water. So in this tetra broth, it consists of nutrient broth. Addition to that, in nutrient broth, they will be adding sodium thiosulfate, calcium carbonate, iodine solution, and phenol red. This particular broth will be enriching the salmonella and shigella. Next is selenite F broth. In that, they will be adding sodium selenate, peptone, and lactose will also be added. This particular broth will be inhibiting the growth of coliform bacteria, at the same time promoting the growth of salmonella and shigella. Next is alkaline peptone water. In this particular broth, we will be adding a peptone, NaCl, and water. So this particular media or liquid media is an excellent medium for growing this Vibrio cholerae or some other Vibrio cholerae present in the fecal sample. Okay, at last we will be seeing few anaerobic media. The first type is thioglycolate broth. So here they will be adding a yeast extract, casein hydrolysate, pancreatic digest, glucose, L-cysteine, agar, sodium chloride. Additionally, they will be adding sodium thioglycolate. In addition to that, they will be adding rizazorin or water. So why this particular rizazorin? Sometimes they will be adding a methylene blue also. So this particular rizazorin will be acting as a oxidation reduction potential indicator for that particular medium. And since this sodium thioglycolate is a reducing agent, and it will be creating an anaerobic environment. That's how the anaerobic microorganism will be growing in this particular broth. So this process is generally employed for growth of anaerobes. At the same time, it is also employed for growing some other microaerophilic and fastidious microorganism. Next is RCM Robertson's cooked meat broth. So here they will be in the nutrient broth, they will be adding a pre-digested cooked meat of ox heart. Okay, so then it is used for culturing the anaerobic bacteria. At the same time, it is also used for preservation of stock culture of various aerobic bacteria. So next we'll be seeing about few agar media. Like first type is simple medium. The example is nutrient agar. So nutrient bath, in addition they'll be adding 2 to 3% of agar. So it is used for generally a complex medium used for routine laboratory work, and this medium generally supports the growth of non-fastidious bacteria. Next type is enriched media, which include blood agar, chocolate agar, and LSS. Okay, blood agar, in the nutrient agar, they will be adding a sheep blood, like 5 to 10% of sheep blood will be added. So it is in addition to the enriched medium, it is also acting as an indicator medium. So it's generally employed for isolating the Streptococcus pyogenes. Okay, next is chocolate agar. So this is a heated blood agar. So they will be heating this blood agar for 2 hours at 55 degrees Celsius. And this chocolate agar is generally employed for culturing fastidious bacteria like your Haemophilus influenzae, Neisseria, pneumococcus. Okay, And next is LSS. So here, as I mentioned you earlier, in the nutrient bath, they will be adding a serum from ox, sheep or horse. In addition to that, they will also be adding a glucose. And this LSS is specifically employed for culturing this Corynebacterium diphtheriae. Okay, the third type of this particular agar medium is selective medium. The example is DCA, deoxycholate citrate agar, and Thayer Martin agar. So this deoxycholate citrate agar, in that they will be adding nutrient agar. In that nutrient agar, they will be adding a sodium deoxycholate, sodium citrate, lactose, and neutral rate. Okay, so this DCA is generally employed for isolation of your salmonella and shigella. Next is Thayer Martin agar. So here they will be taking a blood agar base hemoglobin, and in blood agar base they will be adding hemoglobin supplements, like, and also they will be adding few antibiotics like colistin, nystatin, vancomycin, and trimethoprim. Okay, and this Thayer Martin media is generally employed for selectively isolating your Neisseria species. At the same time, here specifically they will be isolating Neisseria only. So they will be adding many antibiotics like cholecystine, nystatin, vancomycin, and trimethoprim. So for totally 4 antibiotics has been added. So the main role of this antibiotics is that they will be inhibiting the most organism except this particular Neisseria. So that's why it is also called as, it is coming under the selective media category. At last, this indicator media. So example is McConkey agar. So here in this McConkey agar, they will be adding a peptone and sodium taurocholate, agar, neutral lead, lactose. Okay. So here it is generally used for isolation differentiation of lactose fermenting and lactose non-fermenting microorganisms. So this McConkey agar is called as, also coming in a selective media, because here they will be adding, in this media bile salt will be added. So that will be inhibiting the non-intestinal bacteria. Okay, and also lactose is also added along with the neutral lead. So there you can able to distinguish your lactose fermenting and non-lactose fermenting. Okay, and this McConkey agar is indicator media, selective media. Additionally, it is also coming under differential media. So in this McConkey agar, they will be adding a neutral red, and since it is a differential media, why it's called as differential media means the pH indicator will be turning into red in color when the sugar in the medium lactose is fermented. So when lactose is fermented means the pH will be turning into red in color. So due to the production of acid, the pH will be lowered. So that's why the media, the neutral red, will be changing into red or pink color. So other example of enriched media include this Bordate Genco agar, which is used for the isolation of Bordetella, which is responsible for causing whooping cough. Next is LSS. This is used for isolation of this Corynebacterium diphtheriae. Next example is Dorset egg medium. Of course, in this medium, in this particular medium, they will be adding a freshly beaten egg. Okay, so it is generally used for cultivation of your Mycobacterium tuberculosis and Corynebacterium diphtheriae. Next, we will be seeing the example of selective media. These are the list of example. First is this deoxycholate citrate agar. So here deoxycholate will be added, which will be acting as a selective agent for the isolation of this dysentery bacillus. Okay. Next is Wilson Blair brilliant green bismuth sulfate agar medium. So here they will be adding a selective agent which is used for isolation of your typhoid and paratyphoid bacilli. Okay. Next is LJ medium. It is used for the isolation of this Mycobacterium tuberculosis. Next is Thayer Martin medium, which is used for the isolation of this gonococci and meningococci. Next is potassium tellurite medium, which is used for the isolation of diphtheria bacilli. So this DCA is used for isolation of dysentery bacteria, and potassium tellurite is used for the isolation of diphtheria bacilli. Next is this TCBS, with this thiosulfate citrate bile salt sucrose agar. This particular agar medium, selective medium, is used for the isolation of this Vibrio cholerae. Okay, so the next thing is regarding to the indicator medium. So in the indicator medium, so the example of indicator medium, so this Wilson Blair medium is also, it is a selective medium. Additionally, it is also coming under the category called indicator medium. Okay, so in this particular medium, they will be incorporating a sulfate in them. Okay, so what it is generally used for isolating of the Salmonella typhi. So this Salmonella typhi will be reducing the sulfite present in the medium to sulfide in the presence of glucose. So the colonies of the Salmonella typhi will be having a black metallic color. Okay, so from that they will be easily isolating the Salmonella typhi. Okay, sorry, this should be small t. Here I had given them in capital letters, so forgive me for that. Next is potassium tellurite. So this particular potassium tellurite is a chemical which will be added in this particular McCoy Leod medium. So McLeod medium, and this particular potassium tellurite will be reduced to metallic tellurium. So by this diphtheria bacillus. So only this particular bacilli have the capacity to reduce the potassium tellurite into metallic tellurium. At this time, once it has been reduced, the colonies will be having a black color. Okay, so this potassium telluride, which is present in McLeod medium, will be reducing to metallic tellurium by diphtheria bacillus to produce black colony. Next is this McConkey agar. So this agar is generally used for indicating the lactose fermented bodies properties. So the lactose fermenting bacteria will be producing pink color colony, and non-lactose fermenter will be producing a colorless colony due to the presence of this particular neutral red, which is acting as an indicator. Next is differential media. First step is blood agar. So blood agar is both differential medium, at the same time it is also coming under enriched media category. Blood agar is generally used to distinguish between hemolytic and non-hemolytic bacteria. So hemolytic bacteria like Streptococcus, Staphylococcus, which are isolated from throat, will be producing the clear zone around the colony because of the hemolysis, that is red blood cell destructions. Okay. Next is this Naglus medium. So this medium is generally used for identifying the lecithinase activity. So it's generally employed for isolating this Clostridium perfringens. Okay. So this particular lecithin will be added in the medium. So most commonly egg white will be added, which is an abundant source of this particular lecithin. Okay. So those bacteria which have the capacity to degrade the lecithin will be producing this particular enzyme. So from that they can easily isolate this particular Clostridium perfringens. Okay. So the next thing, McConkey agar. Again, McConkey agar is coming in a differential. In addition to that, it is also coming in a selective media and indicator media category. Okay, it has peptone, meat extract, sodium chloride, bile salt, lactose, and neutral red indicator. Okay, so first bile salt will be inhibiting the growth of gram-positive bacteria. Okay, next some bacteria like those or lactose fermenting bacteria will be fermenting the sugar, and it will be producing acid. This will be decreasing the median pH. As a result, the neutral red indicator will be giving a pink color to red color colony. Only lactose fermenting will be giving a pink color or red color colony. Okay, so here I had given you example of lactose fermenter, non-lactose fermenter, and late lactose fermenter. So some organisms which ferment this lactose very lately are called as late lactose fermenter. Example of lactose fermenter include E. coli, Klebsiella. Next example of non-lactose fermenter includes Salmonella, Shigella, and Proteus. Next example of late lactose fermenter includes Shigella shoni. Okay. Next is transport media. The examples include Stewart transport medium and Ames transport medium, which is used for transporting samples that are containing this gonococci. Okay. Next is this buffered glycerol saline, which is used for transporting enteric bacilli. Okay. Next is this Pinkies medium, which is used for transporting the nose and throat swabs, which are containing the media microorganisms like Streptococcus pyogenes, Staphylococcus, and Haemophilus influenzae. Okay. Next is this Carey-Blair medium, which is used for transporting the fecal sample containing the suspected to have Vibrio cholerae and Campylobacter. Okay, so next is this Venkatraman Ramakrishnan VR fluid, with the pH of the particular fluid is 9.2, and this particular fluid was initially used for transporting the feces from the suspected cholera patient. But now this particular VR medium is currently not used. Instead of that, the World Health Organization has recommended to use this Carey-Blair medium for carrying the sample from the suspected cholera patient. At last, alkaline peptone water, the pH is 8.6, and it is used for isolation of this. It is used for carrying the samples which are suspected to have Vibrio cholerae. So from cholera patient, they will be taking the sample and they will be storing in this particular transport medium. In this transport medium, only the growth of the medium will be stored, but at the same time, the media will be preserved. Okay, so while transporting the media, the microorganism should not face any stress. So that's a reason why these kind of transport mediums are being employed. So thank you friends, thanks for watching this video.